is my distinct pleasure to introduce Celine de Jong. And she came, I want to tell a little bit of history, and then I'm going to pass it over to Amanda. Celine, I first met in 2017 when she came to Moss Landing and did her AAUS class. She was an undergrad at UC Santa Cruz and came and, let me go to the slide. There she is, for the group, back when Big Creek, before that big storm, washed out the river. But there's Celine, and doing her science diving class that she went on to do undergraduate work underwater. And in the upper right-hand corner is, for those of you that are science divers, can I see hands? So you remember the field neuro. This is Celine getting a field neurological test. And I thought that maybe before she starts or after she finishes, we should do one on her. She's good to go. But she started at Moss Landing, and she came in to help work with the dive program and also to work with Amanda and I as a graduate student. And she has worked with the dive program, contributed to a lot of different classes, even did underwater pumpkin carving on the lower right, one of the rituals of passage in our science diving class. She also took subtidal ecology. And on the far left picture is a picture of her going down to Catalina Island, where she first saw these rhodolith beds, I think, the first time, with a group from San Francisco State. And I had a couple other students that had worked down at Catalina working on these coral and algal beds that she's going to talk about. And so it's been a great tradition for a number of students to work off the coast of California together collectively down there, and Celine followed in their path, but asked a really interesting question that these beds are ecologically interesting, but very few ecological interactions have been explored, and so she really has done some fundamental and really fantastic work on looking at nursery habitat roles. She went on, I'm gonna let Amanda switch to the next slide. There she is. Talk about Celine's transition. We've been both fortunate to work with her in the field. She's an amazing both researcher, very organized, very helpful in different dive classes. And between Shelby Penn and Celine, they forced me to understand Google Drive. Besides her amazing science work, she also has been really helpful in a lot of different aspects to the lab, both as a student, a lab mate, and part of the staff for a little while. So congratulations, and I'm going to pass it off to Amanda.
Oh, thank you. It has been such a pleasure to work with Celine, and especially I think working as a team with Celine and Diana. I think one of the really cool things from these photos is that we both got to go out in the field and work with Celine. And so there's Diana and Celine diving here in Moss Landing, and this is Celine and me on a boat down in Catalina. But actually, if you see the unifying feature of this, it's actually Matt Hess. So I think that might be an important secret extra. Extra committee member. No. I also, so after that trip to Catalina Island with Karen Crow's group in San Francisco State, Celine enrolled in the subtitle ecology class and headed down to Catalina again, and did actually an incredible amount of preparation and field work and lab work down there to study the white urchin, which you're going to learn so much about. I'm actually not going to focus that much on that. That's Celine. But to study the interactions between white urchins and rhodoliths in rhodolith beds. But it was also a really fun trip, and Celine worked extremely hard with her group. There's the class and Scott down there. I think they were out, on land getting a surface interval in this. She also perfected a lot of skills. You saw the underwater pumpkin carving. She's very good at underwater selfies as well. And in fact, any kind of image like art, I'm sure many of you have seen her Canva art, but she also was really good at getting us to pose. We've got an ML there. And actually, she even took a selfie with an endangered species. She shared this, so pretty neat. This was up in Washington, is that right?
British Columbia.
Oh, British Columbia, yeah. But yeah, so while in Catalina, she worked extremely hard, both in the water, so you can see all the different types of equipment. She's going to talk about the experiments that come from this, but I think what sometimes can be lost is the incredible amount of work it takes just to build and design all of these things. And that was what she did during the daytime, but then at night, Celine could be found. I found this, Celine actually filmed this, which is great. This is her measuring hundreds and hundreds of urchins, which you're going to see the data from, but that's what that looks like. And so just an incredible amount of work day and night in Catalina. And then she came back and attended a seminar by Bruno Pernet, who is joining on Zoom, and he is also on Celine's committee, and really got interested in larval settlement and how and whether rhodoliths might be involved in inducing settlement from these white urchins. And so thus began Celine's second life, foraying into larval biology. And so here she is spawning a variety of animals. So these are purple urchins, which we have locally. She also, you'll see a lot of her spawning white urchins in her talk, so I'll skip that. But it also led to a collaborative project with Luke Gardner, Andrew Kim, and several of us here working on Pycnopodia, that sunflower star larval settlement hues as well. So the skills that she developed really made her an expert, and her expertise was really great to see applied to different taxa. She also was instrumental during her time here at MLML in many other ways. So she was really important to the Deepwater benthos cam project. Here we are out on the John Martin, and she's doing deck work, deploying and recovering the benthos cam. She also was a TA for the invertebrate zoology class, where we celebrated the 34 phyla of invertebrate diversity, half of which are vermiform. I did include it. That means worm-shaped. And so really, really valued there. And she also left a lasting contribution to the Marine Lab when she first started, actually. Before she came here, she worked with the hydrus, and she was involved in a digitizing project with coral reefs. And the corals that were used in those 3D models, actually, you can see in the library in one of the displays, and Celine made that happen. And she also got some really neat footage from when they were filming some outreach about the project, which is just fantastic. And I think the thing that she will leave here for a long time is her legacy on all of us. So I sent an email out asking people for photos of Celine, and there was an outpouring of photos of Celine with others through her many artistic pursuits. She was a big part of the Knit and Crochet Club, and very involved in open house, sometimes bringing Jason to events as well. Thanks for coming, Jason, and family as well. And I think just, yeah, has been a really important personality and presence in the lab. So with that, I will stop because we want to hear from Celine. Please, everybody, let's welcome Celine to the stage for defense.
Okay, thank you, Amanda and Diana, for such a lovely introduction. A lot of those pictures I actually haven't seen before, and brought back just a flood of incredible memories that I've had over the past 4 years here. Thank you all for coming, both in person and on Zoom. I'm really excited to share with you today the culmination of my work here at Moss Landing Marine Labs over the past several years, where I investigated the influence of Rhodolith algae on the life history of the painted urchin Lytechinus pictus. And before I even begin to tell you the story of urchins and rhodoliths, I'd like to introduce a few ecological concepts so you have some context in why this work was even done. And the first being foundation species. Now, by definition, foundation species are recognized as those which create or modify physical habitat, ultimately influencing the structure and the function of an entire community. We see these species across all of Earth's biomes, whether that be freshwater, terrestrial, or marine. Now, foundation species influence their environment in several different ways. First and foremost, they create habitat and provide physical substrate for other organisms to live on or within. And by creating these stable environments, what often follows is an increase in species richness and biodiversity within their ecosystems. In large quantities, they also buffer against environmental stress, such as storms or temperature fluctuations, which help ecosystems recover from disturbances. They also contribute to nutrient cycling, increasing primary productivity and supporting food webs. And they offer refuge for smaller or more vulnerable species, reducing predation pressure. So really important. We've also recognized that foundation species have the potential to create habitats that serve as nurseries. Now, nursery habitats differ from general adult habitats primarily by their function. Nurseries provide essential resources that promote the growth and survival of juvenile organisms before they transition into adult habitats. And how they do this is by increasing food availability by providing abundant resources specifically influencing rapid juvenile growth, by increasing settlement and survival. I'll touch on settlement in more detail in just a moment, but this is to say that these areas are consistently receiving incoming juveniles that ultimately contribute more to the eventual adult population. And by connecting ecosystems. Nursery habitats often act as ecological links supporting species that later contribute to offshore reef or deep sea populations. And what I really want to emphasize here is that nursery habitats support growth and are critical areas for the maintenance of populations. Okay. So now that we have a little bit of ecological background, I'd like to introduce you to a small but mighty alga, the rhodolith. So rhodoliths are free-living calcareous red algae. And what that means is that rather than being soft and pliable like we think most algae are, rhodoliths form by slowly secreting layers of calcium carbonate over time. I've actually brought some rhodoliths with me today to pass around, so you're more than welcome to take a look. They bear uncanny resemblance to those nerd clusters, so don't get them mixed up. But this secretion of calcium carbonate is really similar to how corals grow, which is why you may hear calcareous red algae also being referred to as coralline algae. But they do photosynthesize and require sunlight to grow. Rather than being attached to the seafloor, like most seaweeds are, rhodoliths roll around loose on the ocean floor. They're kind of like little tumbleweeds of the sea. And when there are many rolling around together, they can densely aggregate and form habitats that are known as rhodolith beds. These beds support many other marine fauna in areas they otherwise would not be found, and thus are foundational systems. Rhodolith bed habitats are really unique in many different aspects. Firstly, they're found all over the world in tropical waters and in Arctic waters and occur in depths of more than 270 meters. For those who use freedom units, that's about 880 feet. They also are incredibly slow growing, often gaining just a few millimeters per year. But despite that, they're very long-lived. For example, based on radiocarbon dating, there are estimates for rhodoliths in the North Atlantic showing potential ages of several hundred years. Unfortunately, like many marine systems, they're under threat by multiple environmental and anthropogenic stressors, which include anchoring and mooring. So here we see the effects of boat mooring chains on a rhodolith bed in Santa Catalina Island. These heavy mooring chains will drag along the seafloor and essentially crush the rhodoliths into fragmented pieces. Coastal development is also a risk to them, increasing sedimentation and the potential to smother these beds. Climate threats, such as ocean acidification, increasing temperature, and increasing storm frequency can disturb these beds, and they are also at risk by trawling and dredging practices. But because they create habitat, rhodolith beds encourage biodiversity in otherwise barren, soft sediment regions. So this figure comes from a paper by Gabar et al., where they assessed the impact of structural loss of rhodoliths on abundance, diversity, and stability on benthic communities. And we can see that across all functional groups, taxon richness or diversity in these groups increases much more in areas where there is healthy live rhodolith versus areas in crushed rhodolith sand. You can see that algae attached directly onto these rhodolith nodules, and there's a great diversity of invertebrate life that we were able to see down in Catalina and in all of these beds. Prior research investigating the ecology of rhodolith beds have also shown that these habitats offer services that are aligned to those of nursery habitats. And this includes increasing juvenile abundance. Here are two different studies that looked at the densities of juvenile scallops and their association with rhodolith substrate, and both found greater densities associated with live, healthy rhodoliths compared to other substrates by a significant degree. Additionally, rhodolith beds offer predation refuge. So in this plot, from the same paper by Gabara et al., we see that densities of in-faunal invertebrates, and these are invertebrates that live within the rhodolith nodules, are much denser in live rhodolith areas than in crushed rhodolith sand. The TLDR is basically there's lots of really great places to hide and encourage growth. Rhodoliths also often host a diverse microbial and algal community, which contribute to the local primary production of an area. Epiphytic algae, detritus, and organic matter get trapped within the rhodolith beds and serve as food sources for herbivorous juveniles. Again, another plot from that same paper, we see that the percent cover of macroalgae increases across all different kinds of macroalgae in rhodolith beds, and not so much in crushed rhodolith beds. We're sensing a theme, yes? And then this growth of macroalgae on these rhodolith beds also leads to ecosystem connectivity with increased food availability, as rhodolith beds are also known to serve as foraging grounds for predators from adjacent systems. So we can see in this figure that common kelp forest fishes will roam around in beds in higher densities, most likely because there is increased food availability growing on this substrate that are from kelp forests or other ecosystems that are nearby. Now, coincidentally, a lot of these plots from the Gabara et al. Paper are from rhodolith beds that are located on Santa Catalina, which is off the southern coast of California, and where a lot of my fieldwork took place. I think all of my fieldwork took place there. As Diana and Amanda had mentioned before, back in the spring of 2022, I had the opportunity to join Dr. Karen Crow of SFSU, who at the time was our visiting scientist here at Moss Landing, and her lab to a trip to the USC Wrigley Institute. And it was during this time that I encountered my very first rhodolith bed in Isthmus Cove. And underneath this buoy, I dove my very first rhodolith bed and noticed all of these little white urchins inside the substrate, on top of the substrate. They were littered everywhere. They looked like little bits of popcorn. And this urchin species is the painted urchin, or the white urchin, Lytechinus pictus. Lytechinus pictus are typically found from Baja to the central coast of California. We have seen them here in Monterey Bay. There have been sightings at Hopkins of Lytechinus showing up, especially during warm water events. But this urchin is primarily a southern species, so do occur in warmer waters. They can be found in kelp forests, seagrass beds, and sandy bottoms and rocky reefs, and evidently rhodolith beds. And they're not a very big urchin. I think they get about approximately 5 centimeters in test diameter, but other than that, they're pretty small. The painted urchin, Lytechinus pictus, is a really ideal species to study for 2 reasons. First, they have a significant influence on benthic community structure. They sit at a mid-trophic level and serve as grazers and prey for fish and larger invertebrates, and this includes commercially valuable species like the California spiny lobster and California sheephead. And second, being like many other marine invertebrates, they are broadcast spawners, meaning that they release their gametes into the water column and experience external fertilization. And once that happens, they exist for a period of time as planktonic larvae before they settle to the seafloor. So this brings me to the importance of larval settlement. We know that the ocean is this vast three-dimensional space, and many marine species have what's known as a bipartite life cycle, where they experience two very different forms during their lifetime. And this is akin to a caterpillar turning into a butterfly. As larvae, their sole job or responsibility is to survive, grow, develop, and disperse. And then at a certain point, they'll go through this transition known as settlement, where instead of staying in the three-dimensional open water space, will settle down to the seafloor and metamorphose and turn into the spiky little urchin that we all know and love. And as a juvenile, again, their job is to survive, grow, and mature, and hopefully reach adulthood, where they can then reproduce, and the cycle happens all over again. I bring about settlement as a really important transition or part of this cycle, because this is really where the maintenance of populations is, oh my gosh, sorry. Settlement is basically how we describe this transition from the planktonic to the benthic life stage. This process is really critical for population replenishment, there we go, and understanding species distribution, since it links reproduction to adult population dynamics. And without successful larval settlement, recruitment into adult populations would be reduced, leading to shifts in community composition. There are a lot of questions that surround this process of settlement, but a big one is really, what is it that drives this process? How does this happen? And there are typically two major components that drive the process of settlement, and those are cues and competency. So here is a diagram that outlines the life cycle of Lytechinus pictus, where you can see as a fertilized zygote, it walks through. It grows up as an echinoplutei and continues until it reaches this mature late 8-arm phase, where there's an adult rudiment. And this adult rudiment structure is essentially where the new body of the urchin will form. But how they are able to make that jump from this 8-armed pluteus larva to a fully fledged urchin is through the detection of settlement cues. And settlement cues are environmental triggers, either chemical or physical, that initiate settlement. But during this time in the water column, the larvae has to grow and eat and basically gather enough energy that they can make it through the metamorphosis process. And when they reach that developmental benchmark, we call that larval competency. And this is basically the time where they are biologically ready to settle and are able to respond to cues if they are present. What metamorphosis looks like really stems around this adult rudiment structure. So this is a diagram of metamorphosis in the Pacific sand dollar. You can think of it as basically a squash or flattened sea urchin. But there's that 8-armed larva in the adult rudiment, and it essentially flips itself inside out, where the spines and 2 feet will emerge from the adult rudiment, and then they'll shed their larval skeleton, and you have a bona fide juvenile. Now, what's really interesting is that there have been a lot of studies looking at how different species of coralline algae, including some rhodolith-forming species, induce settlement in marine invertebrates. And there are a number of mechanisms through which this can occur. Probably the most common way is through biochemical signaling. So coralline algae will emit organic compounds such as polysaccharides, proteins, and other metabolites that can then be detected by competent larvae that they then use as that transition indicator. And they can be as general or species specific, depending on which animals and algae are making contact, essentially. Physical structure can also have an influence on settlement. So surface texture and the microtopography of certain coralline algae can provide attachment points and trigger larvae to settle. Biological interactions, such as the growth of microbial communities on the surface of coralline algae or biofilms, that are made up of algae or bacteria can intermingle with these chemical compounds that algae secrete and cause settlement in some benthic invertebrates. And then also environmental conditions. So if temperature reaches a certain coolness or warmness, or if there's enough light, that'll also trigger some coralline algae to produce more of that chemical that larvae can then detect. However, there have been very few studies that have looked at settlement potential in rhodolith species in the context of nursery habitats. These studies typically look at isolated laboratory experiments where they just expose a bunch of different coralline algae to larvae, but not really so much to look at nursery potential, but more just is there a response and what the chemical response or chemical cues might be. And so it was with this knowledge of not knowing so much about the context of nursery habitats in coralline algae triggering settlement and the observations of the urchins that I saw while in Catalina that led me to develop my 3 overarching objectives. The first being to characterize size, frequency, and density distributions as a function of live rhodolith cover. Basically what this means to say is how many post-larval urchins are in rhodoliths and how big are they? My second objective was to assess the preference of post-larval urchins for live rhodolith substrate. Basically, do post-larval urchins prefer to be in rhodolith beds? Is there a behavioral component happening? And my last objective was to explore whether live rhodoliths enhance settlement and subsequent metamorphosis of larval urchins relative to other cues. Basically, do rhodoliths induce settlement of larval urchins? And if so, how well and how quickly do they? So on to our first objective. How many post-larval urchins are in rhodoliths and how big are they? So almost a year to the day that I had gone down to Catalina the first time with Karen Crow, I came back to Catalina with the subtitle ecology class and ran some subtitle surveys with my incredible class who was there, and essentially took 3 different metrics during these transects. And to sort of set it up, these transects were 10 meters long, and I had 10 quadrats per transect that I measured at the beginning of each meter mark. So this is kind of what that looked like. So I swam down the transect and using my little quadrat, measured the beginning of every meter. This amazing picture by Amanda shows exactly what I was looking at. And within this quadrat, I estimated percent cover of live rhodolith. These beds are really patchy in nature, and so sometimes there might just be all sand, or sometimes it might be a full, dense, healthy rhodolith patch, but oftentimes it's sort of a mix of the two. And this is an example of what we can see, or what I saw down in Catalina. So a lot of the white that you see is dead or crushed rhodolith, and then that dark reddish pink color is live. And we estimated percent cover in 2 different ways. The first was by visual estimate of the diver in the field at that time. And then we also took photos of every single quadrat for later photo analysis, where I put down 100 random points across the image and categorized each point to a certain substrate type. Next, we counted the number of urchins that were found within each quadrat within the living layer. And what I mean by living layer is when you're looking down into a quadrat, sometimes you can see urchins that are sort of tucked away underneath the rhodolith, but it's not a very shallow layer. Here's sort of an exploratory core that we took, and you can see that that living layer can actually be quite thick before it hits this anoxic sand. And so we sort of rummaged through each quadrat until we hit that anoxic sand and picked out all the urchins that we could find. And these urchins were then collected from these quadrats, put into marked Ziploc baggies that we brought back to the lab and measured to the nearest 0.1 centimeters. Okay. So this is a lot of data that we have. what did we find? Figure 1. So basically, to orient you to this figure, we have percent live rhodolith cover on the bottom from 0 to 100%. And then we have urchin count per meter squared. That's our density that we found. And the circle size represents multiple data points that are in the same spot on the graph. And so the bigger the circle, the more data points. So we had 110 quadrats that we surveyed, and some of them had the same, recorded live rhodolith and the same number of urchins that were found in that. So that's what those sizes mean. I performed a linear regression and found that there was a significant positive correlation with urchin density as percent cover increased. And when you think about it, that kind of makes sense. With rhodolith, we have a lot more places to hide than when you're out in open sand. So this is a really great example of what a sandy quadrat with only about 10% cover looked like versus a 90% cover quadrat. And there was a recorded 1 urchin in this quadrat that you can't see in the image, and it's most likely under that piece of algae that's at the top. And then 10 urchins that were recorded for this quadrat, but only 8 that can be visibly seen in the pictures. Okay, to orient you to this figure, we looked at individual urchin size of all the urchins that we had collected, and plotted this frequency distribution where I have urchin size on the x-axis, and the height of each bar represents the relative frequency of that urchin size. You can notice in the legend that anything that was rhodolith was defined as greater than 10% cover, and anything that was equal or less than 10% was considered sand. And so here, these dashed lines represent the mean size of each distribution. It can be kind of hard to see when you don't have those dashed lines, but through analysis of a KS test, we did find that there was a statistical difference in the distributions, and that urchins were generally larger in areas where there was rhodolith. This figure is another way of showing kind of the same thing, but instead of looking at individual urchin sizes, I took the mean or average size of all the urchins within the quadrat and plotted them against the percent live rhodolith cover. And again, with the linear regression, we found that there was a significant positive correlation between average urchin size and live rhodolith percent cover, although slightly less so than the first figure that I showed. So we found a really strong association between urchin abundance and the size of these urchins to live rhodolith cover. Larger urchins in live cover suggest that rhodoliths continue to provide resources to urchins as they grow, which may lead to higher survivorship within the beds. The surveys that we were able to conduct only counted urchins that were greater than 3 millimeters, or that 0.3 centimeter mark. But on an aside, we did collect a bucket of live substrate and kept it in the wet lab undisturbed, so we didn't bubble any air, we didn't add that much water. And after a while, we could see these really teeny tiny urchins starting to crawl up the side of the bucket, which was really incredible because these are urchins that we weren't able to quantify or even observe in the field. And the presence of these tiny urchins sort of further highlights the potential nursery role that these rhodolith beds may have if they're such small individuals that are not seen, but seen later. And so essentially, these surveys showed that with increasing live rhodolith cover, we had increasing size and increasing density, which is great, but why? And so my second objective kind of digs further into sort of these potential drivers that might be influencing why we see more urchins in the beds and possibly size. So to recap, my second objective was to assess the preference for live rhodolith substrate by post-larval urchins. And this is to say, is there this behavioral choice where they're choosing to go to rhodolith over sand? And the way that I did this was through the use of subtidal cages. Yay! I've spent my entire scientific career avoiding subtitle pages. But here I am, alas. Essentially, I installed 9 subtidal cages in a sandy patch that was adjacent to the rhodolith bed that I did my surveys in. And over 1/2 of the benthos of the cage, I added live rhodolith substrate that I'd brought over in a bucket from the bed with all urchins and big pieces of macroalgae picked out. And once those were set, I added 6 urchins in a row on that margin between the substrates and let them roam around for about half an hour to 45 minutes. It was really depending on the diving constraints. After which, I went back and recorded which substrates the urchins were found on. Now, there was significant preference of urchins to rhodoliths. So I ran 3 different trials, and they were all subsequent trials. So same urchins that were reset in the same spot. But we found that in trial 1, 87% of the urchins chose rhodolith. In trial 2, 77% of urchins chose rhodolith. And in trial 3, 72% of urchins chose rhodolith. And this is really exciting to see. With some really fancy statistics that I can talk later if you're interested. Basically, from this data set, we were able to calculate a 91% chance of these post-larval urchins choosing rhodolith. Now, we know that there seems to be this active choice, and that kind of alludes to substrate composition potentially mattering, but why? I've started to think about, how do urchins benefit from rhodoliths? And although it wasn't recorded specifically as data, I did observe these sort of heaping or dressing behaviors by these urchins where they would move to the substrate and then immediately start covering themselves with either shell fragments or rhodoliths. They wore little hats. And so this sort of indicates an importance in the use of the surrounding material that they're in. And there's less of that material in crushed carbonate sandy areas. Sort of next steps about thinking of decoupling the response driver. These urchins, I didn't starve beforehand, so there might be some kind of drive for food availability, so that's why they're moving to rhodoliths. Or it might just be complexity of substrate. Are they seeking shelter because they're small, and because they're known foraging grounds, they probably don't want to get eaten by fish. But one thing that we haven't talked about yet is the larval side of this story or this equation, and what role might urchin larvae play in actively selecting rhodolith habitat. And so with this in mind, I developed my 3rd research objective, which was to explore whether live rhodolith enhances the settlement and the subsequent metamorphosis of larval urchins relative to other cues. So essentially, do rhodoliths induce settlement of larval urchins? This isn't something that has ever really been looked at before, not for this species and not for the rhodolith species in Catalina. So I was really excited to sort of delve into this. And a lot of this work actually stemmed from inspiration I had after watching Dr. Bruno Pernet's seminar. So go to seminar, guys. It's really important. It could end up at your thesis. But basically, I had to grow urchins. I had to rear urchins. And so I first started by building this larval stirring rack. This design was modified to fit the water tables that we have here in the aquarium room. And this apparatus is really important because it helps mimic sort of this 3-dimensional environment that larvae would experience when in the open ocean. Larvae are really passive, they're plankton, and so they're not very strong swimmers. And so if you don't have this agitating motion, they'll all sink to the bottom and are unable to feed. After that was built and running, I spawned my urchins by injecting them with a potassium chloride solution through the peristomial membrane. That's kind of like the gums of an urchin. And waited for a spawning reaction, either through the expelling of eggs, which you can see in this video, or through sperm, which you can see in the still photo. So once the larvae were fertilized and set in their jars at their appropriate stocking densities, I began to feed them microalgae. And the microalgae that I fed them was a species called Rhodolith Algae. And this is a species that we actually cultivate in-house at Moss Landing Marine Labs. And I helped with cultivating them, like microalgae farming. It's pretty fun. And the feeding process consisted of centrifuging the microalgae out of their growth medium, and then resuspending them in filtered seawater before feeding them out at a known concentration. Now, along with feeding, I did frequent water changes, which during this time I was able to check in on the development of my larvae. So again, we have this figure of the life cycle of Lytechinus pictus. And here we can see what an early six-armed Pluteus larva looked like through the scope. And so we know we're getting closer to that sort of last stage Pluteus, where they might reach metamorphic competency. For my particular experiment, it took about 41 days for this batch of larvae to reach a 75% metamorphic competency threshold across the entire population. I wanted to make sure that the majority of the larvae that I was going to use in a single batch were metamorphically competent, which I then was able to use for my Q experiment. So for the Q exposure experiment, I had 6 different treatments. They'll look exactly like what's on that cake over there, I promise. And the first, obviously, being live rhodolith. I used 0.1 gram blotted weight of live rhodolith, as well as dead rhodolith, as my structural control treatment. So the dead rhodolith were bleached fragments that I then boiled for about 10 minutes in fresh water to make sure they were like really dead. And I actually let them marinate in our flow-through seawater system in the aquarium room for over a week to accumulate any biofilms that might be in the water and attached to the structure of the dead rhodolith. I also had 2 different chemical cues. GABA. GABA is commonly used in aquaculture to settle commercially farmed invertebrates like abalone. And it acts as a chemical mimic for compounds that are found within a variety of coralline algae species that have been identified previously in their role for invertebrate settlement. The second chemical cue I had was KCl, this potassium chloride. It's similar in that it can influence the behavior of larvae by affecting their sensory systems, and by shifting ionic concentrations, KCl could activate receptors or signal pathways that guide larvae to settle on suitable substrates. My control treatment was filtered seawater. There was nothing in there. And then I had a bio-inoculated sand treatment, which was to act as a complementary treatment to my dead rhodolith treatment as a reduced structural complexity treatment. Now with these 6 treatments, I had 8 replicates, and I added 10 larvae per replicate. This is really exciting. Oh wait, no, almost there. So after I added all the larvae in their little wells, I did incremental check-ins over a 24-hour period. And so within the first hour, I checked in at every 10 minutes, and then I checked in at the 2- and 3-hour mark, and then came back the next day and assessed settlement at the 24-hour mark. So that's a really exciting part. This is probably my favorite figure from my thesis, but essentially what it shows is the mean proportion of larvae that settled at the end of the 24-hour exposure period. So this is at the very end of my experiment. This is all of the larvae that reacted to all of my treatments. And basically what we can see here is live rhodolith outperforming all of their treatment cues by a lot. The letters that are above each of the bars represent statistical difference between groups. So you can see this clustering of dead rhodolith, KCl, and GABA, a clustering of sand and filtered seawater, but then you have live rhodolith in a league all of its own. And also, since I was doing incremental check-ins over time, I also looked at the rate of which larvae were being settled from each of these queues. And this plot illustrates the calculated projections for mean settlement of larvae to their corresponding treatment over a 24-hour period. The points that you see are the mean proportions of larvae, again, settled at each check-in time, and the fitted lines calculated are based on those points. And using an ANCOVA, I found that there was a significant difference in the rate of settlement across all treatments within the first hour of the queue exposure. And I say first hour because when you look at the first hour, this is really when the vast majority of the larvae settled. And so I was able to compare slopes, so rate of settlement. And through these pairwise comparisons, it showed that, of course, again, live rhodolith was the fastest compared to all other treatments. Now, we know that live rhodolith overwhelmingly influences larval settlement in Lytechinus pictus. But what's interesting is that it settled significantly more than just the chemical controls. And this might highlight or suggest some kind of additional non-chemical or compounding factor that's influencing settlement. Otherwise, we would have seen equal settlement rate or reaction to the chemical controls. What was also really interesting is that we saw that dead rhodoliths still trigger settlement, the ones that were boiled in fresh water for 10 minutes. And they had very similar levels of settlement response to my two chemical controls. And so this also might suggest that carbonate sand habitat may still be really valuable sources for settlement cues for Lytechinus pictus, and lends support to the idea that even a degraded rhodolith habitat should be protected. So with my first objective, we learned that more and larger urchins were found in the rhodolith beds. In this one bed, Isthmus Cove in Santa Catalina Island. My second objective, I found that post-larval urchins preferred rhodoliths as substrate, so there was a choice. And with my third objective, I found that rhodoliths strongly induce larval settlement, and not only just strongly, overwhelmingly, and at a faster rate. And I want to bring back this idea again of rhodolith beds as nursery habitats. We can kind of get lost in the sauce here, but bringing back sort of why are we even talking about all of these things? The results of my thesis support the idea that at least the rhodolith bed that I looked at is or acts as a nursery for Lytechinus pictus. There seems to be this ecological importance for rhodolith beds as nurseries, and they serve as critical habitats for juvenile invertebrates. But right now, rhodolith beds are very rarely considered in management plans or marine protection policies, and so this really highlights the need to protect these environments, because even if they seem degraded, they may still prove important for settlement and the maintenance of healthy populations. And with that, I have a lot of people to thank. First off, my thesis committee, Amanda, Diana, Scott, and Bruno. I'm going to read off my presenter notes, otherwise I'm probably going to cry. Okay. Amanda, it has been such a privilege to be welcomed into the fold of your lab, a vibrant community of invertophiles who have been nothing short of compassionate and encouraging. See you all. Your constant enthusiasm for the scientific process is so infectious, and it's really an attitude that I hope to carry on into my future work. Diana, the serendipity of meeting you at a time where, I remember this exactly, I bumped into you at Safeway in Santa Cruz, at a time where I sought to further my academic journey was really uncanny. I would never have experienced the magic of science in the underwater world without your instruction and care. and your ability to notice the extraordinary in the ordinary is like such an incredible gift. Okay. Thank you both for challenging me and believing in this work, and helping me grow in ways that I couldn't imagine before starting this degree. And to the rest of my esteemed committee, Dr. Scott Hamilton and Dr. Bruno Pernay, thank you so much for your wisdom and patience. Scott, for believing in my abilities and knowledge during moments when I was overwhelmed, and I'm sure of what to do next. But also, I really valued your mentorship in the field when we were in Catalina, even though you made me do cages, it's fine. Thank you so much. Bruno, I know you're watching. Thank you for inviting me into your lab with open arms, and taking the time to introduce the incredible world of larval echinoid development and all things Lytechinus pictus. Your thoughtful feedback and perspectives were so valuable in the evolution of this degree. I cannot thank you both enough. Okay, funding sources, institutions, and collaborators. I really want to thank Dr. Karen Crow and her fish lab. Without them, I would never have gone to Catalina and experienced this incredible habitat, and I probably wouldn't have a thesis, but that's fine. Would have figured something else out. And USC Wrigley for hosting us both times when I was with Karen, and then also for our subtitle ecology class. To the vibrant Moss Landing community and staff, there are so many people that I would want to thank and can't fit on this slide. But my invertebrate ecology lab members, both current and former, thank you so much, especially Sydney, Jacob, and Keenan. I don't know why I don't have your name up there. Sorry. Incredible dive support team. Matt Hess, I don't know where he is, but I probably wouldn't have half my thesis without him. Jess, Molly, Noah, and Emily. Aquaculture techniques, I have so much to thank for. From Luke, Ava, Andrew, Hallie, and Evan. Most of those people aren't even here anymore. But Andrew's right down the road, so if you need him, you know where he is. Marine operations, John Douglas and Helena. And facilities and administration. Tara, thank you so much for your help on the administrative side and all the paperwork and stuff. It's just super hectic. And your compassion and patience for the students here is just like unheard of. It's incredible. And then, of course, to all my family and friends, there are too many of you to put, again, on just one slide, but thank you so much from the bottom of my heart for supporting me and bringing much-needed joy and reprieve as I navigated this degree. And you probably see a familiar face on this slide. Last but certainly not least, I want to thank my incredible life partner and fiance, Dr. Jason Toy. Jason, I have no words that can truly encapsulate how grateful I am for your love and your support. You constantly remind me how lucky I am to be here, and your care and patience knows no bounds. Whether we're collecting surf perch on a remote island off of British Columbia or getting engaged in Big Sur every day with you is an adventure I never want to end. I really don't think I would have made it across the finish line without you, and for that I'm so grateful. And with that, I'll take any questions.
We see metamorphosis happening before our eyes. Okay, thank you so much. Great job, Celine. I'm happy to field questions. I also forgot to acknowledge we also have family and friends who have joined on Zoom, so we really have a lot of folks on Zoom. Please do feel free to type in your questions, or if you use the raise hand function, we've got folks watching. But while we're working on that, let's start in the room. Does anyone have questions for Celine? And I'm going to bring the microphone to you, so wait for me. Jess, up first.
you killed it, and you did make me cry, so.
Oh, no.
I was right there with you. So this was an amazing amount of work, and you talk a lot about nursery habitat, and so of course I'm going to ask something outside of the scope of your thesis.
Thank
What do you think is like the next step for these guys? Like if this is the nursery,
Right.
once they become adults, like what's next?
Yeah, so there's kind of this ambiguity when it comes to defining what an adult is. This is something that I talked with Bruno about, which is why I referred to the settled urchins as post-larval and not necessarily juveniles or adults. But we saw these little teeny tiny urchins that were still in the rhodoliths. And so that really suggests that they're staying there for quite some time. Sorry, what was your question?
More so just like, yeah, like if this is the nursery, do you think that they're like going somewhere else afterwards? Or like, I mean, you saw large ones in the rhodolith as well. So do you think it's like a whole life cycle being used in this area?
Yeah, quite possibly. I didn't see any urchins that reached that maximum size that we have observed these urchins getting to. But there was also that image that, it was a photo that you took, where you saw Lytechinus in the adjacent kelp forest bed. So there is some possibility that they are migrating out and that this is sort of just their source population, and may branch out. But when I was thinking about this project, this question also did come up, and the idea of trying to track where an urchin goes is just logistically really challenging. They're not very easy to tag, and so if there were some way to just see where they're roaming around, that would be really interesting. But for now, it seems like this is a really important just source habitat that these urchins are in.
Thank you.
Yeah.
Okay, more questions? Yes. Okay. Do you get to ask a question after you defend? No, you do.
That was so good. Thanks. So when you looked at all the rhodoliths and you like grabbed them out of there after doing your density estimates of the Lytechinus in there, and you found all those tiny little babies that like crawled up the sides of the buckets, were you able to quantify and like extrapolate how many more were in?
No, because
area?
that was sort of just a passive observation. I had collected this bucket of rhodolith substrates to use in my white maze experiments, which got scrapped. But basically had them sitting in a water table with just flow through, but like nothing super like agitating. And the next morning I came back in and I looked at the just inside of the bucket, and on all of the walls were these teeny tiny urchins that had just crawled out of the substrate, and that was kind of just really shocking to see. But nothing that I could quantify. It was really just observation.
So like
but
already
kinda,
significant, but then even more so if you
yeah,
like, just
I don't even know really how you would be able to do that other than just sifting through a known quantity of substrate and then counting, and then I don't know. But it definitely goes to show that there are way more urchins in these beds than we were actually able to count when we were in the field.
Awesome. Good job.
You can totally ask a bunch of questions. Okay, Ailish has a question.
Hi, Celine. Great talk. I really, really enjoyed it. I know you were talking about ambiguity with sizing between post-settlement and adults. I'm really curious for your objective too, how you decided which size urchins to use for your substrate preference.
So there is some kind of growth proxy based on laboratory experiments where urchins that were spawned in a lab and observed to grow in like ideal conditions basically have about 0.5 centimeter, like half a centimeter growth per year. So you can kind of think about every half centimeter of an urchin as like 1 year. And the sizes that I used in my caging experiments ranged from 1 centimeter to 2 centimeters. So I had 1, 1.5, and 2 centimeter urchins. And so sort of under this assumption of like this ideal growth rate, those are probably urchins that are about, 2 to 4-ish years old. So not recently settled, if that makes sense. So,
Thank you.
yeah.
Great job, Celine. I was curious if you knew if there was any data sets describing the distribution and abundance of rhodolith beds in general. Like how common and widespread are they?
Yeah, well, there was this map by a pretty seminal paper by Mike Foster that described the general ecology. The general ecology of where these beds are. And they are globally distributed. This paper is from 2002, so there could be more beds that have been found since then. But I'm not sure of any open source data sets that, quantify what these beds look like or what the densities look like. I could have gone into a lot more detail about, the morphology of different rhodoliths. You can actually see on the table over there, there's a bunch of different rhodoliths that Diana brought in. And there are many different morphologies. There's foliose, there's branching, there's the really dense nubby ones. And so these beds all, even though are comprised of rhodoliths and have similar ecological function, the rhodoliths themselves can vary quite a bit.
Do they tend to occur in areas with certain conditions?
Yeah, so that's why boat moorings are such a concern, because these beds, because they're not attached to the substrate, end up congregating in areas where there's not super turbulent water, so protected coves and bays, which is also where a lot of people like to moor their boats, because they're protected coves and bays. So we see a lot of rhodolith beds sort of showing up in these areas where there isn't a lot of major disturbance frequently. And both in shallow coastal waters, but also these beds that are located 270 meters from the surface. So.
Thanks.
Yeah.
Questions in the room? Yeah.
Great job, Celine. I have a question about, these rhodoliths seem to be supporting a wild abundance of these urchins, and you're finding bigger ones and more of them in rhodolith beds, but was there any evidence in the surveys to suggest that, we know echinoderms are ecosystem engineers, right? With long-spined urchins on coral reefs and purple urchins here, is there any evidence to suggest that they might be, helping the rhodoliths out and be, synergistic in that way?
Yeah, there was a thought I wrote in my discussion about the potential influence of bioturbation, because these urchins seem to wedge themselves in and among the rhodoliths, which might help facilitate shaking off sediments that might, that could possibly smother these rhodoliths. Unfortunately, or maybe fortunately, I didn't look at that, because that's a lot of work. But yeah, there is some thought there and some reasoning you could think of bioturbation being a positive influence on rhodoliths. So not only rhodoliths benefiting urchins, but vice versa as well.
Rhodoliths attracting urchins to try to help themselves,
Yeah.
continue to exist. Because they're definitely doing that.
Okay, we've got 2 questions on Zoom. So we'll hop onto those and then we'll see if there are any more in the room. So the first one is from Ben Walker, who writes, "Great job, Celine. I thought it was interesting that the dead rhodoliths had such an impact on settlement. Do you have a sense of why that might be? Perhaps a chemical question remaining, or could it be surface area?"
Yeah, so that's a really great question. So part of the reason why I marinated them, as well as the sand, in the aquarium room to build up that biofilm was because I wanted to decouple whether or not the dead rhodolith fragments influenced settlement just because of biofilms, but that didn't seem to be the case because we didn't have really any urchins at all that settled on the bioinoculated sand. So there is some component that's happening, or some component that is a part of the dead rhodolith that can initiate settlement. And I have a supplemental slide about this. Basically, there have been other studies that have also looked at dead calcium carbonate as a settlement inducer. And yeah, there does seem to be an additional component, whether that is surface area or some kind of lingering chemical that can't just be boiled out, that is triggering some kind of response from these larvae. And so on the left here, we see a study that looked at coral larvae that settled in the presence of living and dead Crestus coralline algae. And then on the right here, this is a paper that looked at the green urchin. It's a northern species. And we can see that boiled Lytechinus, which is a genus of rhodoliths, didn't actually induce settlement. So it could also be entirely species specific. There's a lot to unpack there and a lot more work that I will let another grad student probably take a look at, so if they're interested.
Yeah, thank you. I should say too, I saw an after message that said this was Tallulah's question. Ben wrote, or under Ben's name.
Oh.
Anyway, so then Cheyenne Jarman writes a follow-up. So wrote, fantastic job, Celine, such a cool thesis. I agree. A follow-up to that question regarding settlement, would KCl mimic the chemical cue potentially provided by conspecifics?
Ooh, that is a really good question. I'm not familiar with what chemical cues are exuded from urchins themselves. And this was actually an afterthought once I had finished my settlement cue experiments to add conspecifics to see if even just the presence of conspecifics could induce settlement, but that wasn't something that I was able to get around to doing. With the Pycnopodia work, though, we did try to see if there was any kind of settlement response in the presence of conspecifics, and I believe that there wasn't any. So again, it could be totally species specific, but not something that I actually directly observed or looked at. So.
Thanks. And we actually ended up with one more on the chat before, and then maybe we'll just take any other quick questions in the room so we can let Celine go on to the next meeting. So this question is from Nicole Crane, who writes, firstly, I hope she's waving. Firstly, I hope to see some of you later at the house. Celine can talk about that. And then she writes, Celine, awesome talk and a great study. You are amazing. Again, agree. I am curious about acoustics, whether urchins at different life stages make different sounds. I assume so. And she wrote that in parentheses, not me. And whether that can be used to assess habitat preference by different life stages. Technology around this has gotten pretty good.
That is a really interesting question, mainly because I never thought to think of it. I am not entirely familiar with like sound in urchins, or if urchins do they squeak underwater? I don't know. But I can imagine that the presence of larger urchins that are bioturbating or shuffling around in these beds does cause quite a commotion or a racket that could probably be heard by other organisms. But I don't know how that would influence settlement or the attraction of other urchins into the beds. Yeah, that would be really interesting to take a look at.
All right, any other final questions in the room? Yes.
First, obviously, fantastic job. It's really amazing to see your odd hours at the lab come together and culminate in this presentation. And I'm sorry, I'm going to ask about your cage experiments.
Oh, that's okay.
I'm a microbiologist, so this might be a little bit on the silly side, but I noticed that there was a decreasing percentage choosing the rhodolith bed, and you mentioned reusing the same urchins for the experiment.
Yeah.
Do you think that's a cage effect? Like if you had used, I don't know, for lack of a better term, fresh urchins?
Yeah,
do you
totally. And there's probably some component of fatigue as well. These urchins are being just like constantly shuffled and moved around. And that was something that we did account for within the model. But there wasn't really that much of an influence of that variable. At the end of the day, looking at the model output, there was still very strong probability that urchins would choose rhodolith, regardless. But yeah.
Thank you.
Yeah, yeah, of course, thank you.
All right, with that, I think we should give Celine a moment of break to get some water and then prepare for her post-defense committee meeting. So let's everyone give Celine a round of applause.